Hello, hello everybody. I'm happy to welcome you all to our first talk this semester. It's about neuroplasticity and it's by Abigail Calder. She's a PhD candidate at the University in Fribourg, where she heads a research project on the effects of LSD on neuroplasticity in healthy people. She also conducts research into the adverse effects of psychedelics and their underlying causes, as well as psychedelics' effects on people's values and well-being. And she also joined the ALPS Foundation as a research coordinator. She completed her master's degree in neuroscience at the University of Bonn and holds a bachelor's degree in psychology from the University of Dresden. And now let's welcome our speaker with a warm round of applause.
Thank you. Thank you. It's really a pleasure to be here to talk about psychedelics and neuroplasticity, which is my PhD topic. And thank you again for the invitation. It's really cool to kick off the first talk of the semester. So I like to keep things a little bit interactive. Don't be scared. But I kind of like to know before I give a talk who the audience is and what you might already know about a topic beforehand. So tell me, just raise your hand if you've heard this before today. Psychedelics enhance neuroplasticity. Yeah. Or psychedelics enhance neuroplasticity in humans. Let's make it more specific. So most people have heard this before at some point. Now raise your hand if you believe it's probably true. Still almost everybody. Raise your hand if you're not sure it's true. A couple people, raise your hand if you really don't think it's true at all. Okay, good to know where we're at. So today, the talk I'm going to give, I have to admit, is going to be maybe a little dissatisfying. My hopes is that you'll learn something, but you might also leave the room a little bit unhappy, because this idea that psychedelics enhance neuroplasticity is a very new theory, and we don't know a lot about it. And so for this talk today, I want to actually just walk you through this theory and what its history is, how it came to be, why people think that psychedelics might enhance neuroplasticity. Our story starts with antidepressants, because that's where the theory started. And then I'm going to talk about the whole theory of psychedelics and neuroplasticity, and what does it mean when somebody says psychedelics enhance neuroplasticity. And then actually I'm going to take a step back and take psychedelics out of the presentation for a little bit and just talk about neuroplasticity to give us some perspective and context. What is neuroplasticity in the first place? And then I'm going to step even further back and talk about how scientific theories come to be in general and how you can judge them. At the end of it, I hope you will have learned something, and I hope you'll appreciate how complex this topic actually is, and maybe wince a little bit next time you read a very simplified journal article claiming that psychedelics just enhance neuroplasticity. Okay, so our story starts with antidepressants. I'm going to start back in the late 1990s with one of the great, well, I wouldn't say one of the great scientific moments ever, but definitely in the psychedelic field. There was one of these moments where something surprising happened that changed the way that we thought about something. And this happened with the work of Dr. John Crystal from Yale. And he says, looking back on this years later, that ketamine was really quite a surprising and in some ways shocking discovery. How many people know what ketamine is, roughly? Yeah, okay, great. So back in the 90s, they were doing research with ketamine, and ketamine interacts with a system in the brain called the NMDA receptor system. You don't have to know what that means right now, but John Crystal wanted to know how this receptor system is changed in people with depression. He was not trying to cure depression. He just wanted to manipulate the NMDA system with ketamine and see what would happen, and so he did. He took a very small number of patients, gave them ketamine, measured some stuff, and was rather surprised to find that these patients started to tell him they were feeling a lot better. He thought that was kind of weird. That was not the point of the study, but it was curious, and so he did it again, and more teams did this, and it turned out that ketamine was unexpectedly a rapid-acting antidepressant. So if you give people ketamine, here's a graph from one of the first studies. If you give people ketamine, they start to feel better, or their depression symptoms improve within just a couple of hours. and they might stay that way for a week or so. And this was kind of unheard of. Most antidepressants that were given at the time and that are still given today take longer than that to work. Some people already feel effects within a couple days when they start a new antidepressant, like Prozac, for example. For some people, it takes a couple of weeks. Ketamine was fast, and it lasted up to a week, so long after the ketamine is out of the body. And this was surprising because it was not explainable by the theories of depression that were around at the time. The dominant theory of what causes depression is that it's a deficit in serotonin. Ketamine has nothing to do with serotonin, really. And so it was a surprise. And then, a few years later, something similar happened. Maybe it was a little bit less surprising to the researchers doing it, but I think it was still surprising to everybody else. And that was that some psychedelics were found to have rapid antidepressant effects. Raise your hand if you don't know what psychedelics are. Don't be shy. Okay, nobody. Great. In this case, it was a psychedelic drug called psilocybin, derived from magic mushrooms. This was a pilot study done in London by Robin Carhart-Harris and colleagues, finding that psilocybin also had a rapid antidepressant effect after one dose, combined with therapy in a safe setting. And this effect lasted for a while, at least for some people. So now we've got two drugs, ketamine, psilocybin, that have a really fast antidepressant effect that seems to last, and nobody's really sure why. Not only that, but some of the other psychedelics appear to have rapid therapeutic effects, not only on depression, but on some other psychological disorders. For example, here's a graph from a study that was actually quite recent, studying the effects of LSD on anxiety. So after a dosing session with LSD, I think it was 100 or 200 micrograms, people felt less anxious pretty much right away, and they continued to feel less anxious. And there were also some studies with other psychedelics, including ayahuasca, MDMA, DMT, and a few others for other types of disorders. So what we've got here is a rather unexpected finding where ketamine, psychedelics, also MDMA, have rapid therapeutic effects within hours that seem to last for a while. This is probably not news to most of you. But it's really important for the history of this neuroplasticity theory because, of course, people started to ask themselves, why? Why is it that psychedelics and ketamine cause these rapid effects that way outlast the presence of the drug in the body? And there's one thing I want to comment on here before I move on, because this question, why are psychedelics antidepressant? For some people, the answer is a bit obvious. If you ask the patients, they have a lot to say about that. They'll say, well, it's because I learned something, I understood something about myself, or it can be 100 different things. And I really don't want to discount that. It's absolutely part of the story, and I'll come back to it. But a lot of scientists really want to know what's going on in the brain neurobiologically, and so they started to look for it. And that is how we get to psychedelics and neuroplasticity. So this is the question of why are ketamine and classic psychedelics having antidepressant effects in the brain beyond whatever subjective experience is going on. So how many of you have heard the term neuroplasticity before and could potentially explain to somebody approximately what it is? Yeah, okay. So neuroplasticity is the brain's ability to change its structure and function. And one important thing to remember is that everybody has neuroplasticity all the time, from the moment a brain is formed up until probably even the moment of death. The brain has some ability to change and adapt. You might think of it with the metaphor of a blacksmith's hammer. So if you have a chunk of metal and you want to change its shape, you have to heat it up, and then you have to stimulate it with, for example, a hammer. And using this metaphor, you could think that our brains are always a little bit hot. Maybe some brains are hotter than others or more plastic than others, but they're always a little bit warm, always a little bit ready to be changed. At the molecular level, that means that neuroplasticity involves the growth or alteration of dendrites, synapses, and sometimes brain cells themselves. So here what I have on this slide are neurons, which are the cells in your brain that communicate with each other and lead to experience. And neuroplasticity is actually an umbrella term that involves a lot of different processes. at the level of the cell, even within the cell, but also between cells. It describes the way they change their connections with each other. And so at the cellular level, for those of you who are interested in the molecular biology of this, neuroplasticity means, for example, neurogenesis, so the growth of new brain cells. As the brain grows, of course, it has to grow new cells. In adults, this is much less important and only happens in a few regions, but it's one facet of neuroplasticity. Here you see also a couple of neurons, and do you see a pointy light? No pointy light, okay, then just neurons. So here there are two neurons. This is what they look like. You have the cell body is the big fat part in the middle, and they have branches that they use to communicate and connect to each other. So neurons always have a cell body and these branches. And neuroplasticity involves changes in the way these cells communicate with each other. For example, when cells form a connection with each other, that's called a synapse, and neuroplasticity involves modification of these connections between brain cells. New ones can form, or they can also degrade, or the strength of synapses can change as well. And neuroplasticity also involves changes in dendrites, so those are what we call the branches that reach out to other cells in order to communicate. I explain this because it's somewhat necessary for understanding some of the findings about neuroplasticity, but don't worry if it's a little bit technical. And neuroplasticity also means changes in gene expression. So genes that are important for supporting all of these growth processes become more expressed, including growth factors. At the level of the brain and at the level of your experience, all of these little molecular changes manifest in changes in brain networks and behavior. The most important and well-known correlate of neuroplasticity is learning. So whenever you're learning something new, let's say you learn to play the piano, there's something in your brain that's physically changing. And if you get really good at the piano, it might even be a big enough change that you can see it on an MRI. But neuroplasticity is also important for memory, for brain growth, for healing of the brain after injury, and for any adaptation to the world in the absolutely broadest sense. So any change in the way that you interact with and process the world requires neuroplasticity. So what does all this have to do with depression, right? I started with depression. The point is that in depression, neuroplasticity is severely impaired. And this is something that has been shown in a lot of studies. So people with depression show a sort of atrophy of the brain. It has less of an ability to grow. And just to give you an overview of what that theory is like, one theory of depression is that it tends to start with chronic stress. Stress not in the sense of having too much to do at work, but just any difficult life circumstances whatsoever can raise somebody's susceptibility to depression, as well as a few other things. So your brain is constantly adapting to the world, right? If you find yourself in a new circumstance, your brain will change itself in order to adapt. If you find yourself in a very stressful situation or a very stressful life circumstance, your brain will also adapt. And at the same time, if that stress continues over a longer period of time, the adaptations that your body and brain make are actually pretty bad for neuroplasticity. Inflammation tends to set in, and over time, you can start to lose some of the brain's ability to change. And so what can end up happening is that somebody who's been exposed to a lot of chronic stress has a stress-adapted brain. For example, they might have a lot of focus on the negative, they might kind of shut down and just be a bit in survival mode all the time. And over time, their ability to change that becomes impaired. So even if the stressful situation is over, you can't necessarily get back to normal because of this impaired neuroplasticity. That's basically the neuroplasticity theory of depression. I'm not going to get too much into it, and it's more complicated than that, but just to give you an idea. That's why we even talk about neuroplasticity at all. Okay, so back to the psychedelics. Because we know that neuroplasticity is important in depression, people wondered if maybe these new rapid antidepressants were changing the brain via neuroplasticity. So in the slide I just showed, there were some neurons with some branches. Remember this? On these branches, you have even smaller branches that can grow or atrophy. And back in 2010, this was one of the first studies that showed that ketamine causes the branches on neurons to grow. Not only the big branches, but also the small ones called dendritic spines. And there were a lot more studies on this that confirmed that indeed ketamine enhances neuroplasticity in a few different ways. It makes the branches of neurons grow, it leads to the growth of new synapses, and it leads to the upregulation of genes that are important for supporting these changes. And a couple of years later, the same thing was found for the psychedelics, for ketamine, but also for LSD, DMT, psilocin, even MDMA, and a couple of the others, seems to enhance a lot of different aspects of neuroplasticity. So they make these dendritic branches grow, they make dendritic spines grow, they upregulate the signaling that supports this growth, and overall, this seems to lead to changes in connections between brain regions and changes in emotion and behavior, because we wouldn't care about it if it didn't at some point lead to changes in emotion and behavior and thinking. Here are a couple of pictures from some of these studies. So in the first one with the blue neurons, what you see there is a rat neuron in a dish. The first one on the left has been treated with basically a placebo, so they didn't do anything to it. The second one has been treated with ketamine, and the third one has been treated with LSD, and you just see that they grew more branches. And in the picture on the bottom, same thing. On the left is the placebo, and then you see 3 different psychedelic drugs. LSD appears to be the most effective, at least in this picture. But you see that these small dendritic branches grew after treatment with psychedelics. I also, together with my doctoral supervisor, tried to summarize the timing of these changes, because that's actually really important. Is this an effect that lasts an hour or a week or a month? Because you would want to know that in order to design, for example, therapy interventions that happen in that time. And it seems like, you don't have to read this whole figure, but basically this growth period, this sort of springtime in the brain, appears to last for about 3 to 5 days in these studies of animals. So after they give them, for example, psilocybin, there's growth for 3 to 5 days in which more of these branches grow and more synapses form. And then after that, there's no more growth, but the synapses that did grow and the branches that did grow survive. So what you get then is essentially a growed brain. And these changes were found to survive for at least a month. What you also see in this figure is we divided these studies into human studies and animal studies. So the animal studies results are shown in purple, there's a lot of those, and the human studies are shown in yellow, fewer of those. Most of these findings have been done in animal studies, and the human research on this is still really young, and the results are kind of mixed. Can anybody figure out why, actually? Why? Anybody have any idea why it's harder to research neuroplasticity in humans? I promise it's a little bit obvious. Yeah. Okay, so not really. It's allowed to give psychedelics in studies. Otherwise, I'd be in trouble. Yeah? Precisely. So with all of these animal results, most of them were gotten by taking the brain out of the skull, because that's the best way to look at the brain, is to look at it directly. With humans, that is illegal. You can't really do that. Also extremely unethical. So we have to find more indirect ways to look at neuroplasticity in humans, and it's really difficult, which is part of the reason why the results are somewhat mixed. So basically, most of these findings on psychedelics and plasticity have been found in animals. In humans, the evidence is indirect and a little bit mixed. And this is the theory of psychedelics and neuroplasticity as it stands right now. So the theory goes like this. Psychedelics enhance neuroplasticity via these molecular mechanisms in the brain. In particular, the serotonin 2A receptor for the classic psychedelics and the NMDA receptor for ketamine. Last year, actually, there was a paper that came out showing a common molecular mechanism for this, namely remodeling of the extracellular matrix. But there's really this focus on the molecular mechanism. So you give a drug, and even if it's just cells in a dish, you'll see enhanced neuroplasticity. And what's also part of this theory is that for a few days, or maybe even weeks, after taking psychedelics, the brain has more potential to grow and change. That's the theory beyond this theory. And this, it's hypothesized, is why psychedelics have this transdiagnostic potential to treat depression, but also anxiety and maybe addiction and a few other conditions as well. This is the theory. This is the stand of the research today. and I'm nowhere near done. Because now I want to pull back a bit from the psychedelics and talk a little bit more about neuroplasticity, because it's important to understand what that is and why we care about it in the first place in order to contextualize these findings with psychedelics. So, we actually haven't known about neuroplasticity for very long. For a long time in the history of neuroscience, people thought that the adult brain just stays the way it is and doesn't change. Kids grow, their brains grow, but as soon as you're an adult, you're stuck like that, for better or for worse. And around the 1960s, there started to be evidence to the contrary. This is a picture of Professor Marion Diamond from the United States. I think she was at Berkeley. She was one of the first people to really prove that the adult brain can grow and change. And she did decades of research on this. And she summarizes her work like this. In decades of research, they found 5 items that were essential for building a better brain, for enhancing neuroplasticity. The first was diet. The second was exercise. The third was challenge. The fourth, newness. And the fifth, which they found later, was love, or healthy social relationships. So if you want to enhance neuroplasticity, that means you eat a healthy meal, go for a run with some friends somewhere new. I'm not going to say on LSD. But there's a lot of things that enhance neuroplasticity that have nothing to do with drugs or with psychedelics. And that's what they found in their research. It's a basic function of the brain that evolved to help us adapt to the world. And so there's also quite a bit of evidence for this, which I would just briefly go over. Exercise, for example, enhances neuroplasticity in humans in the ways that we have to measure that. Exercise is also a pretty good antidepressant. It's a lot more difficult, especially for somebody who's really down in their luck, to motivate oneself to exercise. But if you can do it, it does work, and it even seems to work in some meta-analyses better than antidepressant drugs, better than SSRIs. Combining the two works particularly well. And these studies in the 60s that studied plasticity, as well as most of the research with psychedelics and plasticity, have been done on lab mice and lab rats. Raise your hand if you've worked ever in a mouse or rat lab where people did experiments on mice and rats. Yeah, a couple people. Yeah. Me too, man, no shame. I want to take us to some context here, because what I've got is a picture of a standard lab rat environment. I've worked in 3 mouse labs, and they all looked like this. So the mice live in a box that's about this wide and this long. They've got some straw on the bottom, which they kind of like to burrow in. They've got a couple of other same-sex mice, so they don't fight too much. There's some food up top. It's always the same food. And there's a water bottle. And they spend most of the time there. It's basically like mouse prison. So this is the standard laboratory environment, which all of these studies have been done in. That means small cages. There's very little opportunity to move around and exercise. There's almost nothing to play with. There's nothing to do. They don't have any toys or anything like that. They get the same food every day unless there's some reason for a scientist to try to motivate them with something good. And they're with the same other mice every day. It's extremely boring. And you can even find some pretty good evidence that mice and rats don't like this kind of environment. I mean, they are prey animals, particularly mice, but at the same time, that doesn't mean they are, I'm not going to say fulfilled, but that they are living their best mouse life in an environment like this. We had a mouse lab that I used to work in. I don't do mouse research anymore, but we had to clean the cages about a couple times a week. And in order to clean the cages, you have to take the mice out of their cage and put them in a plastic box that's about this high, and then take away all the poop and everything, and then put them back. And there was this one mouse that every time we put it in the holding cage, would start to do these incredible backflips. I'm not joking, it was crazy. The first time I saw it, the cage is this high, and she backflipped this high. And most of the time she would get out, and we would have to chase her down, which I feel terrible, honestly. But you can tell that she had a compulsion to move. Every time, as soon as we put her in the other cage, she's backflipping constantly. And the other mice are also running around. They want to move, and they're in just such a small space. Another fact about lab mice and lab rats is that they're notorious alcoholics. It's extremely easy to get a lab mouse addicted to alcohol. All you have to do is put a bottle with about, let's say, 10% ethanol in their cage with them, and they just start drinking. Why do you think that is? It's the only thing to do. They're bored. There's nothing going on. And, you know, if we were in just a tiny room with the same people every day and nothing to do, and somebody puts a bottle of tequila in there, you know what you're doing. The point is, this environment is not really the environment that a mouse evolved to be in. It's unusual. And somebody noticed this, also back, I think it was in the 60s, and started to study what happens if you change the environment. What happens if you take a mouse and you put them in what they call an enriched environment. It was originally called rat park. So here are some examples of rat parks. You can see they've got a lot of space. They can run around and exercise. They have more friends. They have more toys. There's more stuff to do. I think I see a trampoline up there. And it's just a lot, there's a lot more fun, a lot more things to explore. And what do you think happens if you put an alcohol bottle in this cage? Do you think they turn into raging drunks? Usually not. They actually usually ignore it. Some of them may be a little predisposed, but it's then like 1 out of 10 or something like that. So this is an enriched environment. And if you remember from the previous slide, one of the things that enhances neuroplasticity is novelty, newness, and challenge. So what do you think happens to neuroplasticity if you take these rats from the standard environment and put them in the enriched environment? You see the same changes in neuroplasticity that we've been seeing with psychedelics. And this was found back in the 60s, 70s, 80s. There's a big body of literature on this. So here you see another neuron with its branches, its dendrites. The standard environment is on the left, the enriched environment is on the right. In the enriched environment, the rats simply grow more dendritic branches. Here's another figure showing essentially the same thing. So this one is basically zoomed in on one of the branches from the previous figure. They're from the same study even. And in the standard environment, there are just fewer little dendritic spines. And in the enriched environment, there are more. So there's enhanced neuroplasticity. And you see enhanced neuroplasticity in pretty much all of the ways you can measure it that you have with psychedelics. So it's quite similar to this figure with the ketamine and the LSD. The difference, I think the reason that the control group here is smaller is that these neurons are a bit younger, but you see the same trend, that there's more branching. Same thing here. There was this figure showing that particularly LSD enhanced the growth of dendritic spines. But these animals were in a standard environment. You know, to my knowledge, there haven't been any studies with psychedelics where animals were not in a standard environment. So if you take one from a standard boring cage and you give it LSD, this happens. But if you put it in a more interesting cage, something really similar happens. Both of them seem to enhance neuroplasticity. So where does that leave us with psychedelics? I really want to zoom in a bit more on these 2 points that were found in this research, this seminal research on neuroplasticity. They found that newness and challenge were both extremely good for neuroplasticity. They both fertilize the brain, so to speak. That means new experiences, new environments, and new challenges upregulate neuroplasticity. And you can probably think why this would happen, just evolutionarily, why the brain would evolve to do that. Because if you're in a new environment or something new and surprising happens, you need to adapt. The brain is going to upregulate its ability to adapt. That would be extremely useful. It may even be that strong challenges, and I don't mean challenge in necessarily a negative sense, it could be a positive challenge, may create something called pivotal mental states, which is actually a psychedelic-related theory describing how the brain induces rapid learning after stress or radical changes in the environment. So if you are, for example, a rock climber, I love rock climbing metaphors with psychedelics, bear with me. If you're a rock climber and you try something new, a new challenging route where you can learn something, you might have a slightly more plastic brain afterward. And here's the point. Psychedelic experiences themselves are nothing if not new and challenging. And I don't mean challenging in the sense of bad trips. I know challenging experience is sometimes used to mean something like bad trip. I mean challenging in the sense that it asks something of people. People are quite tired when they're done with their LSD trip. Psychedelic experiences are new and challenging. They're so novel, especially the first couple of times, that it's very hard to describe to somebody how the trip is going to be. I know because it's my job, and I fail at it every single time. I like to ask people after they're done with their LSD trips if it was as they expected. Nobody ever says yes, at least if it was their first time. It's new. Everything's new. So there have been no studies of psychedelics in these enriched environments. It's all been in the standard environments. And it's possible that the mice in these standard environments are starting with a deficit in neuroplasticity compared to a wild mouse, compared to what they've evolved to be. So that means it's possible that this psychedelic-induced neuroplasticity is particularly visible when there's a deficit. And indeed, in humans, the most robust evidence of psychedelic-induced plasticity so far comes from ketamine studies in depressed people. And that's mainly because the research on depressed people has been going on for longer. It's been going on since the late 90s and not since 10 years later. And with them, you also see some signs of upregulated neuroplasticity, particularly a few weeks after treatment. So it's very hard to find these rapid changes. But still, the most robust evidence comes from animals in standard environments and depressed patients. Healthy humans, you might say, live in human park. We have a lot to do, we have a lot of friends, we have a lot of different foods, a lot of things to adapt to. And so it could be that these changes in neuroplasticity that you see in the healthy rats don't translate very well to healthy humans. It's possible that there is some effect, but this really throws a wrench in the works with the theory of psychedelic-induced neuroplasticity, particularly because in these studies of neuroplasticity in mice, the researchers found a ceiling effect, meaning you can only enhance neuroplasticity so much. There's a level above which it won't go, probably because at some point it becomes dangerous or somehow detrimental. So this is one wrench in the works, and actually there's two. Firstly, novel experiences enhance plasticity. That makes this theory of psychedelic-induced plasticity more complicated, because originally, a lot of this data came from animal studies where they do really mechanistic research. They look at cells, they look at neurotrophic factors, so they are really focused on the molecular mechanisms. But neuroplasticity is not just molecular, and that's been known about plasticity for decades. Novel experiences also enhance plasticity. That, by the way, is extremely difficult to research in humans because you would have to take a human and put them in a completely new environment. You'd have to move them to another country and then randomize who has to move to another country and who doesn't. It's just very impractical. Actually, psychedelics may be one of the best ways to study that. But psychedelic-induced plasticity, there may very well be a molecular effect, particularly because you find it in cells in a dish who presumably are not having a particular experience, although I wouldn't be 100% sure about that. But there's also probably an effect of the fact that it's a novel experience, and an interaction between those two things, between the molecular effect and the experiential effect. So that complicates things. And secondly, these lab animals on which this theory is based might actually have a neuroplasticity deficit compared to the wild versions of those animals, which might mean that the findings, it's just not clear how well the findings translate to healthy humans, particularly healthy humans. Okay, are you dissatisfied? Confused? Well, I often end up talking about science toward the end of talks. I like to zoom out a bit and get to the bigger picture. And I hope I've given you a good idea of how this theory developed. I hope I've been able to kind of walk you through where it came from and why people think psychedelics might enhance neuroplasticity and what we still don't know. And so now I'm going to get to scientific theory in general. And keep in mind, these things that we've been talking about are relatively new. Neuroplasticity was discovered in the 60s, basically. Ketamine-induced plasticity was found, I think, in 2010. It wasn't much earlier than that. Psychedelic-induced neuroplasticity really hit the scientific field 6 years ago. 6 years ago. It's not very long. And yet, you see this idea that psychedelics enhance neuroplasticity everywhere. You see it in all sorts of papers. I've put it in papers. It's me too. You see it in journal articles. I saw it on Netflix. And it's only 6 years old. And when reading scientific papers, I'm not going to lie, it can be really difficult. How many people in here are either studying a scientific field or want to work in a scientific field at some point? Quite a few, yeah. Which means you have to read research papers. Which means you have to somehow judge the quality of the evidence in a research paper. And that's really, really, really difficult. It's not always clear how good the quality is in any given paper. You almost have to be an expert even in order to judge that. And so scientific theories you could think of as a sort of Jenga tower. Does everybody know the game Jenga, where you have to build up this tower of stones and then you try to pull a stone out that's not going to make the whole thing crashing down? So in the context of science, some theory that's at the bottom of the tower is a theory that has a lot of evidence that's really robust. So this might be something like psychedelics are therapeutic for some people some of the time. Something that's pretty much on solid ground. I feel like I can say it and no one's going to fight too hard on it. And then as you go up the tower, you get theories that are on weaker and weaker evidence bases. You could also say some theories are built on a foundation of solid rock and some are built on a foundation of sand that could just crumble away at any moment. Where do you guys think this theory of neuroplasticity is on the Jenga tower? There's no right answer. I don't know the answer. But it's not at the bottom. And it's probably not at the very top. Where would you put it? Would you put it in the middle? This is the part where I could use the pointer. It's not always easy to judge, but the tower might look something like this for the field of neuroplasticity. So there's quite a bit of evidence that depressed people tend to have reduced neuroplasticity. That's maybe toward the bottom. Psychedelics enhance neuroplasticity in healthy rats. There's some studies showing that. Pretty much every study that's looked for it has found that, but there's not that many of those studies yet. Then this idea that psychedelics are therapeutic because of neuroplasticity is a decent theory, but it hasn't been proven at all. It's just a theory. And then at the top, you might have something like psychedelics enhance neuroplasticity in humans, and that's why they're going to cure Alzheimer's disease. How many of you have heard that somewhere before? Yeah, okay. Thankfully not so many, because it's probably not true. So you can see how basically on the basis of a theory like psychedelics enhance neuroplasticity, people build and build and build and publish, and then magazines publish what they published and maybe tweak it a little bit, maybe make it a little bit more clickbait friendly. And it's hard to judge where you are on the tower when you're reading something. I don't have time to judge where every single paper I read is on the tower. But I think it's important to keep in mind, because you see this idea that psychedelics enhance neuroplasticity everywhere. And I think if you remember one thing today, it actually shouldn't be about psychedelics or neuroplasticity. Particularly for those of you who want to go into science, if you remember one thing, remember, don't get overly attached to your theories, because they might die, and then it'll feel like a part of you died. I'm going to take like 2 minutes to talk about myself and how I started my PhD. So I started my PhD and decided with my supervisor on this project where we're going to test whether LSD enhances neuroplasticity. And I kind of thought that the evidence base for this was pretty good. Otherwise, I maybe wouldn't have done it. And I started reading up on research papers that were supporting this theory and suggesting this theory. And I kept seeing that people were citing things in a way that they maybe shouldn't be. I'll give you an example. There's this paper that was published in 2013 by, I think, Catlow and colleagues. There's nothing wrong with the paper itself, just to be clear. But they gave rats, or mice, I think it was mice, psilocybin. So some mice got a placebo, some got a low dose of psilocybin, some got a high dose of psilocybin. And then they measured neurogenesis in the brain, so the growth of new neurons. In the placebo group, there was not really any difference. In the low dose of psilocybin, there was no statistically significant difference. There was a very small increase in neuroplasticity, but it could have been random because it wasn't significant. So basically no difference. And then in the third group, the high dose group, they found a significant reduction in neuroplasticity. So after the mice got psilocybin, there was less neurogenesis, and that was statistically significant. I've seen this paper cited in a lot of different reviews. Guess which finding they talk about? Yeah, they don't talk about the high dose psilocybin usually. They talk about this potentially random slight increase in the low dose psilocybin group and say nothing about the significant decrease in the high dose psilocybin group. And I could go on with a lot of examples like that. So this is how you might know that someone is getting overly attached to a theory. People start cherry-picking information. Eventually, this made me mad enough that I wrote a review and tried to also incorporate a lot of the null findings and the complexity. So this is how you can tell if you are going into science or if you're reading scientific papers, having encounters with scientists or discussions, that someone's overly attached to their theory, which in psychedelics can happen relatively easily. First of all, there may be a lack of curiosity about opposing views. So maybe even lack of acknowledgement that the high dose was bad for neuroplasticity in that particular example. There may be kind of an uncritical acceptance of theories that are at the top of the Jenga tower. I've seen it cited in a few places that psychedelics might cure Alzheimer's disease, which is based on one or two theoretical papers that laid out the theory for why that might happen. And there's nothing wrong with those papers, but people read those and take them as something they're not. And I remember I was at the ALPS conference a couple of years ago. It might have even been the very first ALPS conference. And somebody in the audience asked a panel of neuroscientists how they thought psilocybin could help Alzheimer's disease because he'd been giving it to his grandma with Alzheimer's disease and hoping that it would help. Yeah. And the panel was a little struck, I think. They were polite, but struck, and basically said there's zero evidence for that, which is true. So there's this whole ecosystem around science where, for example, if there's a review paper like that or a theory paper, probably some magazine picked it up and turned it into something it wasn't, and then somebody read that uncritically, which is not even their fault because nobody has time to be critical all the time, and then started dosing their grandma with psilocybin, which, by the way, can have negative effects on the heart. So in older people, you've got to be kind of careful with that. So this is probably evidence of being over-attached not only to a theory, but to psychedelics. Because if you really care about Alzheimer's, you'd go in the clinical trial registry and find the most promising drug in phase 3. Psychedelics are in phase nothing for Alzheimer's disease. Then you might also see a compulsion to defend a theory that somebody's overly attached to, because that feels like, it almost feels like you're defending yourself. If you're overly attached to this idea that psychedelics enhance neuroplasticity or that psychedelics cure something, I think that's maybe more common. There may be a compulsion to defend that even in situations where it's not appropriate. I've definitely had situations like that where, for example, you might find an error in someone's statistics that totally changes their results, and then they tweak it so that it doesn't actually change their results. Which brings me to point 4, which sadly is probably more common in science than we want it to be, that people make biased choices about what information to use in their studies or their statistics or their literature reviews. If you dive really, really deep into these topics, you see that bias everywhere. And so for people going into science, try to guard against it in yourself. Just because I thought if I'm coming for a talk, I should show a little bit of new data. We recently, in my lab, published submitted a meta-analysis for publication. We finished it on something called brain-derived neurotrophic factor. So this is one of these growth factors that supports neuroplasticity, and you can measure it in blood. And people think that if you measure it in blood after you give someone LSD and it's elevated after LSD compared to before, that's evidence for enhanced neuroplasticity, right? The findings on BDNF are all over the place. Some studies find an increase, some studies find nothing, some even find a decrease. What tends to happen is when there's an increase, it's talked about a lot. When there's no change, it's barely talked about at all. We found in our meta-analysis that there was no effect whatsoever of any psychedelic or ketamine on peripheral BDNF, despite the fact that BDNF is often touted as evidence of psychedelic-induced plasticity in papers. We just finished this week, and hopefully it will be out soon so people can actually read it and judge for themselves. So that's what I would kind of end with. Don't get overly attached to theories. But because it's so easy to do, I think you also need something to replace it with. And then I would say, get attached to curiosity. Because if you're a scientist and your theory falls, if you find evidence to the contrary, like we kind of did with BDNF, I didn't expect to find nothing, to be honest. If you're really attached to this theory that you have, it can feel really disappointing. It feels like you failed. But if you're attached to your curiosity instead, if you follow your curiosity, then you sort of get a new life. You get to now figure out why everybody was wrong, including you. And if that excites you, you should be a scientist. And especially in the field of psychedelics, I mean, we have a lot of people in this field who are extremely intrinsically motivated. A lot of people for whom this research is personal. I'm no exception. And that has a lot of positive sides, too. There's so many people that are really motivated, that care a lot. I think people at psychedelics conferences are about 5% nicer than people at other conferences. It has a lot of benefits. But I think one drawback is getting overly attached to the ideas that you have about psychedelics. Curiosity has to have a higher priority because this is new territory for everybody, and psychedelics, as we know, are nothing if they're not novel. So, in summary, psychedelics and MDMA and ketamine seem to have some therapeutic effects for some people some of the time in a safe setting, always an asterisk there. Animal studies suggest that they enhance neuroplasticity and that there are some molecular mechanisms involved, so it has to do with how psychedelics bind to the receptor. And that's a pretty good theory. It's worth researching, it's got some evidence for it, but there are some complications. And so next time you hear that psychedelics enhance neuroplasticity, I hope you can think about these. First of all, novel experiences also enhance neuroplasticity. So it could be that the experience of a psychedelic itself can enhance plasticity. And by the way, the less novel that experience is, the less it might also enhance neuroplasticity. There might be diminishing returns over time if people do it a lot. Secondly, remember that lab research, animal research in a standard environment might be happening in animals that have reduced neuroplasticity to start with, and that could change the results. And that might mean that these animal findings are not perfectly translatable to humans, particularly not healthy ones who presumably don't have any deficit in plasticity. And above all, when you're reading scientific papers, doing science, dealing with theories, try to remember where they are on the Jenga tower, and remember to stay curious and not get too attached to theories, because they die all the time. Thank you, guys. References, if you need them, if you can read them.
Thank you very much for your talk. We now have some time for some questions, so you can raise your hand and we will bring the microphone to you.
First of all, thank you very much for the talk. It was really interesting. It aroused my curiosity. You mentioned before that there was kind of a ceiling for the neuroplasticity, and it was dangerous to go above. That's why I don't know what you really meant by that, and I would be interested to have a bit further information.
Yeah, sure. So what they found in these studies with psychedelics, so this was the study from Calvin Lai and colleagues in the Olson lab at UC Davis in the US, they found that basically there was a ceiling effect with the dose. So it's not so that if you give more and more and more LSD that you get more and more and more growth of dendrites, for example. And they also found that, so there's a molecule that I mentioned, BDNF, brain-derived neurotrophic factor. That is what the brain normally uses, among other things, to stimulate neuroplasticity. And if you give them both, you don't get a summation of the effect. It hits a ceiling. I'm not sure people know exactly why that is. There's just an assumption that if the brain puts a ceiling on that, there's probably a reason, evolutionarily speaking. There's also some evidence that hyperplasticity or too much plasticity might be associated with autism and other neurodevelopmental disorders. There's a few papers on that. To be honest, I'm not sure where it is on the Jenga tower. But I think it's just an assumption that if the brain is blocking that there's a reason.
Cheers. I am familiar with a theory that with psychedelics, the dendrites around the auditory system significantly grow, and therefore depressed humans tend to actually feel the music they hear during a journey or a trip much more, and therefore feel for the first time in a very long time something. So therefore that is a theory that depressed people actually get a feeling of life again. So maybe you can help me out. Where does this stand on the Jenga tower?
Yeah, great question. That's a really good point. Where exactly in the brain do psychedelics enhance neuroplasticity? Because it's not everywhere, and you wouldn't necessarily want it to be. So psychedelics, you're right, probably enhance neuroplasticity in the auditory cortex. In fact, that's exactly the research question that we're researching in our lab. We measure it in the auditory cortex and the motor cortex. Yeah. And in general, they appear to enhance neuroplasticity in the cortex more so than anywhere else. Potentially also in the hippocampus and a couple of other subcortical regions like the claustrum. There may be some changes in the amygdala, but the cortex is where most of the changes are happening. You also see that, at least with classic psychedelics, the cortex has a lot of serotonin 2A receptors, which are the receptors with which classic psychedelics interact. As far as the timing, it's quite interesting. I think the studies in animals have not yet zeroed in on the timing enough to answer that question. What they found is that after 1 hour of stimulation, they don't see changes in neuroplasticity yet, and after 6 hours, they do see them. So if you take, for example, an LSD trip, after 2 hours, somebody is already very well on their way to tripping. But it's not clear if this neuronal growth is already happening at that stage. It might happen later. So the relationship to the subjective effects is not exactly one-to-one. There may be some changes in functional signaling that are already occurring at that time, and then the structural plasticity changes occur later. But yeah, I think somebody would need to measure it hour by hour to find out exactly when it appears, but it's probably not one-to-one. You really see this with DMT. DMT also enhances neuroplasticity. In humans, a DMT trip lasts 10 minutes. The changes in neuroplasticity have not appeared at all 10 minutes in. So it's not exactly the same.
Thank you.
Thank you for your talk. I had a question. Is it common in this field of psychedelic to have like a debunk paper? In that sense that, so you said that there are multiple papers following the theory that psychedelics enhance neuroplasticity, but why is there not a paper that proves the opposite? And just like, for sure it's less attractive, but I mean, it can be a proof.
So the question is, why are there not papers showing that psychedelics don't enhance neuroplasticity?
Yes.
Yeah, so how many of you have heard of publication bias in science? This idea that, yeah, positive findings are more likely to be published, negative findings are much less likely to be published. In every paper I've seen that investigated psychedelics and neuroplasticity in animals, they did find some kind of effect. There was, of course, also the study with the neurogenesis where the high dose was bad for neurogenesis, so that's one. It's hard to say if there are papers that are out there that find absolutely nothing and just weren't published. I should hope not. As far as I know, all of the animal papers have found it. Some of the studies in humans have not found it, or they found reductions. So those papers do exist, but it could be that there might be some null results out there that were never published. Yeah.
If the theory is not that attractive, do you still get fundings or not for your research?
You usually apply for funding before doing the project, before you know what the results are.
Okay.
Yeah.
Hi there.
Yeah.
My question is about the role of therapy, because neuroplasticity, so in your theory, neuroplasticity equates a benefit for depressed patients. But is there a notion whereby plasticity without some kind of direction, without going somewhere, without a positive feedback or positive environmental cue that would do something, goes nowhere? So you can have as much plasticity as you want. If you're not actually inducing some kind of reprogramming in a positive sense, then you're not going anywhere.
Yeah.
Is that something, well, could you comment on that? Or I don't know. Yeah.
Sure. Do you remember this metaphor with the sword and the anvil and the hammer? So the sword heats up more, so to speak, if there's more neuroplasticity, and the hammer is what forms it in a particular direction. Importantly, there's always a hammer, unless you just sit in an empty room for a week after taking LSD, which would actually also probably change your experience. So the experiences that you have in, let's say, a few days or a week after psychedelic therapy would be more important than usual. They would be more impactful than usual, according to this theory. That's why it's important to know how long the enhanced neuroplasticity lasts, because in that time window, there may be more of an opportunity for therapy to be helpful. It's a really interesting question if psychedelic-induced plasticity has a tendency to cause particular changes without therapy. I think it might have a tendency to cause particular changes and not be completely random, even if there's no therapy, and that's because of where in the brain it's happening. A lot of the effects of psychedelics are concentrated in the cortex, as I mentioned. Enhanced neuroplasticity has been found particularly in the in the frontal cortex, which is responsible for emotional regulation. So more growth in that area might mean that this part of the brain has more ability to regulate other areas of the brain that produce emotion, which would cause a tendency, maybe just a small one, for there to be positive effects on emotional regulation even without therapy. But still, experiences matter a lot, and I think you're much more likely to get the change you desire if you combine it with something that is promoting that, like therapy. So in all of these studies with psychedelics, there is some kind of therapy or support that's combined with it for safety reasons, but also because it probably just is more effective. It's also more effective with other antidepressant treatments, by the way. If you combine therapy with exercise, which enhances neuroplasticity, or with other antidepressants, which also enhance neuroplasticity just more slowly, then it's also more effective. Experience matters.
The way that I understood how psychedelics work before was that parts of the brain communicate that didn't communicate before during the trip, but not after. Is this also a form of neuroplasticity, or?
Yeah, so you can divide into functional and structural neuroplasticity. In the talk, I talked a lot about structural neuroplasticity. That means the literal structure of the cells is growing or changing. But functional neuroplasticity describes how cells communicate with each other, how these synapses are firing with each other. So under psychedelics, there's quite a complex effect on that. Some brain regions communicate more than normal. Some actually also communicate less than normal. There's just a change. It's an altered state. You can't really say that it's generally more or less. And you could say that that's a form of short-term functional neuroplasticity, yes.
We have time for one more question.
I have actually two quick questions. Does it matter what kind of psychedelic we're talking about? MDMA and 5-MeO-DMT are not the same. Intensity of the experience, does it matter or not? And secondly, where on that scale that you talked about is, what are some concrete problems that can be solved if there is indeed strong evidence for this happening? CTE or like Alzheimer's, what else could it be?
Yeah, well, you already mentioned one of them is, are there certain drugs that are better at enhancing neuroplasticity than others? This paper that I mentioned, it's on here, Nardau toward the bottom, and colleagues from 2023, they actually tested in either mice or rats how long enhanced neuroplasticity lasts with different drugs. I think they tested LSD, MDMA, psilocybin, and either ketamine or DMT or both. Basically what they found was the longer the substance effect was, the longer the window of plasticity. So in this study, LSD was the longest, which made me happy. And then psilocybin was a bit shorter, DMT was even shorter than that. However, this is just one paper on a couple dozen mice. You shouldn't believe anything based on a couple dozen mice. It needs to be replicated. So it's a bit high on the Jenga tower. But that is one very important next problem, is comparing drugs. Are there certain drugs that are better at it? 5-MeO-DMT is actually a bit of a special case because it has a bit of a different pharmacology than the other psychedelics. It acts more so through serotonin 1A receptors than 2A receptors, which might actually mean that it has different effects on neuroplasticity. It might affect different regions more than others. But yeah, that's a very interesting question, is comparing drugs. And then another avenue of research, which I didn't mention but probably should have, is that there are some researchers who are trying to develop drugs that can enhance neuroplasticity without the trip. And for psychotherapy, you can see why that might be even undesirable for some people, because people say they learn from the trip, and if a bunch of patients say that, I tend to believe them. But for other indications, like Alzheimer's disease, for example, or stroke patients, people with neurodegeneration whose cognitive function might even be impaired, they don't necessarily need an acid trip. And so these non-psychoactive psychoplastogens is what they're called, might have potential there if they're able to enhance neuroplasticity based on the molecular mechanisms without the trip. And speaking of this attempt to dissociate the trip from the molecular mechanisms, I think that's also a big question right now. How much of this is the experience and how much is the molecular mechanism? It's really, really hard to disentangle those. There was one recent study that did that with ketamine, and they gave, I think, another anesthetic while people were on ketamine, and they found no difference between groups, but that other anesthetic may have also had an effect, so it's really, really difficult. But yeah, those are just some. And also, how long is the window of neuroplasticity? I think that's a really, really important question for humans, for human research.
Let's give one more round of applause for Abigail's great talk.